So cell respiration is the topic today, and I'd like to talk about the Krebs cycle, or also known as the citric acid cycle, or Krebs citric acid cycle. Again, another tongue twister. And this is Hans Adolf Krebs, who discovered the citric acid cycle, and as a matter of fact, he won the Nobel Prize for this discovery back in the 1950s. I think it was 1953. Well, so what I'm going to do now is I'm going to assemble the Krebs cycle. And you should have watched also the video on the oxidative decarboxylation of pyruvate, or the link reaction, because that is now a continuation. And look what I got behind the picture here. Surprise! This is going to be, the Krebs cycle that I have to assemble. And we're going to take it again step by step, okay? What do you remember? What is the end product of the link reaction? Yeah, it's acetyl coenzyme A. And this is the substance that we now are going to completely break down to release energy. So I have to find acetyl CoA, and I've already got it over here. And I'm going to move everything else over here to the side. And I have to sort everything out a little bit here so that we can assemble the Krebs cycle. And this acetyl CoA, the acetyl group contains 2 carbons, and this acetyl CoA is now going to be completely broken down to carbon dioxide. And the first step is a very important one. What we're going to do is we're going to take the C2 substance, acetyl CoA, and we're going to combine it with a C4 substance to make citric acid. Okay, and this is why the cycle is called the citric acid cycle. And of course, citric acid must have 6 carbons. Why? Because 2 plus 4 is 6, obviously. So we're going to assemble it. No, that's the way it goes here. Over here we have the a 4-carbon substance, and we need a small little arrow somewhere over here. And we're going to combine the 2-carbon acetyl coenzyme A with a 4-carbon substance here to make citric acid. But notice there is an arrow also going away here. And this arrow that goes away, that is the group coenzyme A, which we have to remove again. Okay, and it goes back then to the link reaction where it's used again. So you see, as I told you already before in the previous video, this coenzyme A, is simply used temporarily, and it's removed here again, and it goes back. Yeah, so that is basically the first important step, and you have to know the name citric acid, because as a matter of fact, this is the substance that gives the Krebs cycle the name Krebs citric acid cycle. Okay, and it is also the first substance that can be identified in the Krebs cycle. So what next? Remember what I told you just a few minutes ago, that we have to completely break down the acetyl coenzyme A, which is now attached and is not present anymore, but now became part of citric acid. Well, in order to completely break it down, we need to remove carbon dioxide. And look, what I'm going to do now is the first step is an oxidative decarboxylation reaction. This sounds familiar, right? So when we have an oxidative decarboxylation reaction, we are removing carbon dioxide. So we're going from a C6 substance all the way down to a C5 substance. Okay, that's the first step. And what do I have to do? Well, oxidative decarboxylation means I have to remove a carbon dioxide, and I have to oxidize the citric acid, and that means I have to remove NADH. I'm simply going to now make one arrow, and I'm not going to make a separate arrow for carbon dioxide and NADH. I'm just going to use one arrow simply for the sake of simplicity. So this is our first oxidative decarboxylation reaction. Okay, so we are have removed already 1 CO2. Now, what do you think is going to happen now? Look, how do we go from a C5 to a C4 substance? What do you think? What do we have to do? What about another oxidative decarboxylation reaction? Okay, look, here's another one. Yeah, over here, CO2 and NADH again. Yeah. So, I need to move over a little bit, otherwise I'm blocking my own picture here. You know what, I'm going to just move everything over a little bit here. So that's better. Yeah. So we have, after we made citric acid, we have 2 oxidative decarboxylations, and then the energy is present in the form of high energy electrons in the NADH. We added 2 carbons over here. We're removing 2 carbons over here. Yeah? And therefore, we're back to a 4-carbon substance, but it's not the same one as the one over here. Okay. Now look at this here. A huge arrow, and there are arrows going away here. And this is where we get out a lot of energy. And the first source energy that we get out is directly usable ATP. That's pretty nice. Yeah. You know what? I have to move everything over a little bit here so that you see this better. Okay. Yeah. Yeah. So the first thing that we get out is directly usable ATP. That's very nice. Yeah, because after all, ATP does carry a lot of energy. And look, I have 2 more substances over here. One of them that you already know. One of them is NADH, and the other one is known as FADH2. And that's the second one that comes out. Now, what in the world is FADH2? FADH2 is also an electron carrier. So just like NADH, it carries high energy electrons. It's simply a different substance. This should not worry us, but the role of FADH2 and NADH is the same. And then of course the last one is over here. We're getting out again NADH. Yeah, and then we're back to the original C4 substance. Okay, and the cycle is complete. In reality, the Krebs cycle is way more complex, but I think these are the key milestones or the key things that happen. And one of the things that you have to always take care of is that you really trace the correct number of carbons. That's what we've been doing, okay? And the second thing that is really important is by asking yourself, what happens to the energy? Where did the energy go? And the energy is now present in the form of high energy electrons in NADH here, in NADH here, in FADH2 over here, and in NADH here. And of course, over here, we also have directly usable energy in the form of ATP. And the Krebs cycle is finished, okay? So my suggestion here is the following, when you have biochemical pathways to draw like this, use your common sense. Don't just simply memorize it, but you remember, okay, if I have a C4 substance with a C2 substance, 4 plus 2 is 6. I must get a C6 substance. And it happens to be citric acid. I do a step of oxidative decarboxylation, and this means I'm dropping down by 1 carbon. I'm dropping down by another carbon over here. Okay. Yeah. So you see, it's by using a little bit of common sense, you don't have to simply blindly memorize it, but you're able to reconstruct what is actually happening. Yeah. So you know what I have over here, somewhere all the way on the other side of my table, of my desk, I actually also have a little textual summary of what's actually going on here. Okay, I'm just going to carefully place it over here, the Krebs cycle, and these are the stages here. If you just remember those 4 lines over here, then you're able to reconstruct the Krebs cycle. Acetyl-CoA is a C2 substance combines with a C4 substance to form citric acid. First step. 2 steps of oxidative decarboxylation, here and here, now covered up by the piece of paper. ATP formation down here. And again, oxidation, just a pure oxidation without decarboxylation, okay? Pure oxidation by the formation of FADH2 over here and NADH, again covered behind the sheet of paper. Huh? Yeah, and if you remember those 4 stages, then you're able to reconstruct the Krebs cycle. Huh? So, I messed it up now. Okay, but the next step that we have to talk about, again, in a different video, is how can we convert this NADH here, this NADH here, this FADH here, and this NADH over here, all of those things which contain high energy electrons, okay? This energy has to be converted now to ATP. And that is what happens in the so-called electron transport chain or in oxidative phosphorylation. Again, that is a different video, but for right now, the Krebs cycle is finished. Again, practice drawing this. The more often you draw it, the more easy it will become. And if you just simply memorize those stages over here, then you're very easily able to reconstruct the Krebs cycle. Okay, but for right now, I wish you all the best and see you around in the next video. Bye-bye.